Today we're going to be talking about zinc. Zinc plays numerous important roles that all plants need to perform key essential functions. In this video, we're going to be breaking down exactly what zinc does in your crops, how it's taken up by your crops, and the soil dynamics that influence how easily we can pull zinc from our soil so our crops can use them. The first example we're going to talk about is gene regulation for growth hormones. The plant is an orchestra with each of the different instruments playing their own role. Zinc acts as kind of the invisible conductor, telling certain roles to turn on and off. So gene regulation is basically a toggle, and zinc is the nutrient that flips that switch. When we turn on or off these switches, it's letting the plant know to release certain plant growth hormones like auxins and cytokinins. Auxins and cytokinins are at the tips of the growing points. So that could be at the tip of the shoot where we have new growth coming, or at the tip of the root. And these auxins and cytokinins let the cells know to elongate and divide and continue to grow and expand, allowing our plant to grow. A zinc finger protein is what binds to the DNA of the cell, and the cell in its DNA is kind of like an instruction manual on what to do. And so zinc will bind to that DNA and allow it to express itself, basically saying, this instrument needs to play its part right now. And zinc binds to the DNA that has those instructions, so the plant knows that it's that specific process's turn. So zinc makes sure that each of the processes in the plant are orchestrated and working together harmoniously. And when we don't have enough zinc, we don't have the ability for those zinc finger proteins to form and bind to the cell's DNA, and we kind of lose coordination in the plant, and therefore we lose a significant amount of efficiency because the plant's not doing exactly what it needs to do at exactly the right time. The second key function zinc does in a plant is carbohydrate metabolism, sugar use. Zinc is responsible for taking the sugars leaves make through photosynthesis and turning them into stored energy, and then turning that stored energy into usable energy later. So in photosynthesis, we have CO2 and water and sunlight, and that gets turned into sugars. Now, the CO2 is entering into the leaf through the stomata, the little holes in the leaf that let that leaf breathe. When the CO2 comes in, it's kind of like a bunch of cars going onto the highway. And we have a maximum efficiency, which is all of the cars can move at the same speed continuously onto the highway and everything goes without a hitch. Then sometimes we have backups, we have traffic jams with that CO2 coming into the leaf. The reason is that CO2 needs to be broken up into a bicarbonate. That CO2 needs to dissolve in water. Remember, we have sunlight, CO2, and water. We're going to use that to create sugars. That CO2 needs to get broken apart. That happens pretty slowly, and we end up with that traffic jam that we talked about. So that plant created this enzyme called carbonic anhydrase. And what that does is it allows that CO2 to dissolve at 1,000,000 times the speed, turning that CO2 into a usable form so we can create sugars. And guess what nutrient plays a key central role in this carbonic anhydrase enzyme? Zinc. So if we don't have enough zinc in our plant, we get a massive traffic jam of all of the CO2 entering into the leaf. And then our plant's photosynthetic efficiency drops, meaning the amount of sugars we're able to make exiting the roots to feed our microbes, to create this engine of soil life so our plants can grow as efficiently as possible, all of that drops. That's why zinc is so important for photosynthesis and sugar creation. Now we're going to talk about something that doesn't happen as often, but is still super important. When our soil is waterlogged or compacted, all of the oxygen gets used up. If we didn't have a backup plan, all of those cells would die. And so what the plant created is this enzyme called alcohol dehydrogenase, which guess what nutrient plays a key central role? Zinc. Now, alcohol dehydrogenase allows the cells to continue to use energy, continue to breathe and stay alive under short periods of waterlogged and low oxygen environments. So if you have a very wet spring or heavy rains that create an environment that is anaerobic at the root zone, so your roots are growing into a soil that doesn't have any oxygen left in it because it's waterlogged, we use this alcohol dehydrogenase enzyme to continue creating energy so those root cells don't just immediately die. Now we're going to be talking about zinc uptake. Once the zinc goes from that soil solution into the plant, it gets taken up to where it's needed, and then it is immobile. That means it can't be redistributed throughout the plant from the older leaves to newer leaves, meaning we always need a constant supply of zinc from the roots into our plant. How does zinc go from an ion in the soil to be taken up by the roots so that it can be used all around the plant with all of those key functions we just talked about? Zinc's taken up in the soil solution in its ionic form, which is zinc 2+. When it's in the soil solution, it is water soluble and it can be easily just grabbed by the roots and taken up. But most of the time it's not in this form. And there are a few key scenarios when zinc turns into what's called an insoluble compound. So it goes out of this form and then is not able to be taken up by the roots anymore. You might have heard of a zinc and phosphorus interaction, especially on the Mulder's chart. You might see an arrow between zinc and phosphorus as an antagonism. The reason for that is zinc has this 2+, and a phosphate ion is an anion, meaning it has a negative charge. When we get a positive and a negative, they stick together and they create another insoluble compound, zinc phosphate, where we have 3 zincs giving us 6 positives, and we have 2 phosphates giving us also 6 negatives. That's 6 positives and 6 negatives stick together, and now that zinc is no longer able to be taken up at the roots. This happens when we have an abundance of phosphorus right at our root zone. Say we did a banded fertilizer application and we have an excess amount of phosphorus right at the roots. A lot of that phosphorus can interact with the soluble zinc and create these insoluble compounds, and now your plant isn't able to access this zinc as well. Another interaction to just keep in mind is in a waterlogged or anaerobic condition, we have sulfur getting turned into an ionic form through anaerobic microbial breakdown, and that sulfur has a 2-, which binds with the zinc's 2+, creating another insoluble compound, this zinc sulfur. So if you have like a rice field that gets flooded, a lot of times you may end up with that sulfur detaching from the soil organic matter, combining with the zinc, and then preventing zinc uptake during critical growth times. So with all of these uptake antagonisms, it's important to note that even if your soil test says you have adequate zinc, we also want to be doing tissue tests to figure out if that zinc is actually getting into our plant. Because sometimes we can have adequate zinc on our soil test, but a lot of it could be in these insoluble compounds and your plant can't actually take them up. So it's important to do these combinatorial tests so we can see the amount of zinc that's in our soil, but then also is that zinc in a form that can be taken up, and we measure that through tissue tests and things like plant sap analysis. Now you're also going to get an increase of available zinc with the more amount of organic life that you have, the more amount of soil microbes, pulling that zinc from being attached to soil particles and making them soluble and turning that zinc back into a soluble form so it can be taken up by the roots. So a couple key pieces to keep in mind to prevent these uptake antagonisms from happening. We generally want to follow a couple rules of thumb, which is our phosphorus to zinc ratio. We generally want to be around 10:1 on a soil test to prevent those zinc phosphates from happening. If our pH is above 7 to 7.5, we get those zinc ions combining with carbonates and creating an insoluble compound. So we may want to look at a chelated zinc supplementation through a foliar application or a soil application in a chelated form, which is going to bypass those insoluble compounds because it's complexed in a way where it can't immediately form those interactions with carbonates and hydroxides. It's also important to be aware of that zinc is a cation. So a cation means it has this 2+ charge. And you may notice that calcium, magnesium, potassium also have this plus charge. And that means when we have a soil particle that has a bunch of negative charges, this zinc is going to attach to it. That process is called adsorption. And the way that we get this zinc off of the clay into the water-soluble form that our plants can uptake is through when the root produces root exudates that acidify and break this bond and turn this zinc ion back into its water-soluble form that the root can uptake. That process happens through root exudates with the acids generated from the roots and through microbial activity that produce acids as well to separate this zinc ion from the clay particle. To recap, zinc is the switchboard controller or the orchestrator telling the plant when to turn on and off specific genes so the plant knows which functions to turn on and off at different times. It's also important for controlling carbohydrate metabolism, sugars going from being made to being stored to being used, turning those sugars into each of those forms. Zinc's uptaken in the soil solution in its ionic form, that's zinc 2+, but at higher pHs or in the presence of antagonistic nutrients, it can form insoluble compounds that prevent uptake. The two key ones we want to look at is high pH, turning into the zinc carbonates or zinc hydroxides, and then with heavily banded phosphorus fertilization or excess phosphorus above a 10:1 phosphorus to zinc ratio, we can create zinc phosphates, which again are insoluble and therefore not able to be taken up by the plant. If we encounter those specific conditions, we want to look at using a foliar zinc or a chelated zinc to supplement the zinc that we can't really get from those insoluble compounds. If we don't have enough zinc, our plants can't turn the sunlight and carbon dioxide into sugars as efficiently, and we end up with a traffic jam. And zinc's job is unblocking that traffic jam and making sure that everything functions as efficiently and smoothly as possible as we turn sunlight into sugars and then sugars into energy for the plant to use. Again, my name's Jack. I'm the founder and CEO of Cultra, a nutrient analytics platform where we let you take all of your lab tests and drag and drop them into our software so you can find those imbalances. Figuring out if you have enough zinc in your soil but it's not getting to the tissue. Maybe it's because your pH is too high, maybe it's because your phosphorus level is too high. We help find those imbalances and show them to you so you know exactly where to put your fertilizer dollars and where to put your effort in order to maximally increase the efficiency of your farm.